So with that, I think I think that was all the announcements I had, and I guess we'll just start. So I want to introduce our first speaker, Tony Vine. He's a professor from my alma mater, Purdue University. So it's always great to have a fellow Boilermaker. It's online. We had Gary Bergstrom yesterday. He's a fellow Boilermaker, and it's funny, you know, Tony and I never met. So I left. I graduated in 1998. And Tony came that same year from the University of Guelph, and we just missed each other, I think. I left and you came on board, and so we never have met in person. But he does some fantastic things. I want to read some of this stuff. You know, he's a fellow. He's not far from here, from Ontario. So he's you know, grew up just north of our border here. So he's probably a little familiar with this part of New York. Did a lot of his, you know, his work at Guelph, and then transferred again in 1998 to West Lafayette to Purdue. He has, you know, several graduate students that he works with, and the research focused on understanding interactions of tillage, crop rotation, plant density, and nutrient placement systems, and the physiology of the crop responses. Current investigations include corn hybrid and plant density comparisons at multiple nitrogen rates to better understand kernel component changes during reproductive growth that will lead to higher yields and higher nutrient recovery efficiencies. And today, you know, you might be intrigued by Tony's title, you know, the keys to tricky big corn. And originally had just big corn. And Tony says, well, there's a lot of things that, you know, we could talk about getting big corn. He says, but I want to talk about the tricky ones. So Tony, I'm going to leave it to you to share your screen and tell us about the tricky keys to big corn.
I'm going to try again here.
I'll let you know when I see it.
Host disabled attendee screen sharing. So we're going to have to try a different route here. Can the host enable me to share my screen?
Hey, Brandy. She's on the alignment. She's right next to me though.
Okay.
Tony cannot share his screen. Tony, what did it say again?
It's, oh, now it's coming up.
She just said you're good. You're good now.
We're able to share now. So
all right, you should be good. There you are.
Oh, for that opportunity. And,
I see you.
Put it into gear here in a second. So are you seeing the full screen, I hope? So, okay.
Yep.
Very good. Well, it's a pleasure to be able to talk to you this morning about some tricky keys to big corn. Before getting into the subtitle, I just wanted to say a few things. Number one, I would much rather be there in person, like Mike emphasized earlier in his introduction, and perhaps we'll have that opportunity someday at a future Corn Congress. Number two, the biggest news in corn is the very rapid increase in price that we've received since last fall. A very unusual event in the sense of having that much of an increase in price during the harvest period and the early storage period, you might say. And so that, of course, is prompting more discussion, you might say, more thoughtfulness, perhaps, about how to take advantage of these, thankfully, higher prices than we've seen for several years. And so the approach in the subtitle here might be, well, we could approach it by perhaps having bigger plants, assuming that the grain yield as a function of the plant weight stays the same. We could approach it by perhaps having more kernels or perhaps bigger kernels. And so in my discussion today, I'm going to deal with all three of those as ways of perhaps approaching the idea of achieving bigger corn. The presentation today is only possible because of the fact that I've always enjoyed teaching, and I particularly enjoy working with graduate students. And so there's 3 graduate students that have joined me and helped to make some of these slides possible today. And they include Leah, Lauren, and Garrett. And I would also like to refer you to a website on which we have more information from our group in terms of news from our group, as well as other resources that you might find interesting. I would encourage you to ask questions, and I'll try to make sure that this presentation isn't so long that I run out of time to answer those questions. But I'm sure that you'll be able to follow up with emails as well. Modern hybrids are uniquely different. And this is just a picture of a 2015 hybrid compared to a 1958 hybrid that were grown side by side at the same plant density of approximately 32,000 plants per acre. And what strikes you about the modern hybrids is, of course, the fact that they have so much more stay green than is the case with the older hybrids. Modern hybrids are also regarded as being more stress tolerant, and those stress tolerances are often evident in terms of better tolerance to drought stress, better tolerance to the stress associated with high plant density. And sometimes the advertisements, for stress tolerance kind of go to the extreme as this picture shows here, with an Aquamax hybrid taken at a Farm Progress show several years ago. But it's also true that not all hybrids are going the route of having progressively bigger plants. And so you've likely heard from the folks at Bayer Crop Science that they're planning to introduce short corn hybrids, which are reduced in stature. And that might be another development along the road to achieving bigger corn a different way. And so time will tell whether that's going to be a progressive strategy in terms of getting to bigger corn, and particularly for those of you that are interested most in harvesting whole plant corn silage instead of just the grain that we perhaps more traditionally think about, and certainly in the Midwest cash crop production environment. There's no question about the fact that modern corn hybrids have been bred in a way that they are effectively achieving higher plant density stress tolerance. And so the increase in plant densities in, for instance, a state like Iowa is changing at the rate of about 350 plants per acre per year in that whole period from 1990. So the last 30 years, we're seeing quite an increase in plant density, and there's no question that that's been a significant increase. But before going too far in this whole thing, I wanted to address the issue of return to nitrogen and how it varies with different hybrids. And one of the fastest ways, I think, of illustrating how hybrids vary quite a bit in their response to nitrogen is to take some of the results from a commercial co-op. And this co-op results that I've borrowed from here from Randy Brown with Winfield United They're under the Land of Lakes umbrella, and they regularly have, let's say, more than 100 locations per year. And in those locations, they are testing hybrid varying from 80-day corn to 120-day corn, depending on where the test is located. And they effectively are always looking at how much variation is there in terms of the response to nitrogen. And so you can see here a situation where in some cases for these 240 hybrids that are shown on this graph, in some cases the response is 50 bushels, and in other cases the response to nitrogen is 115 bushels. So there's quite a wide range in the responsiveness. How are these determined? Well, these are determined by comparing yields for the same hybrids at low nitrogen. And the low nitrogen here is shown at as 0 to 100 pounds, an average of 60 pounds of nitrogen per acre at these on-farm sites. And then the high end is shown here, which is basically a non-limiting rate. And so in most cases, if you're following, let's say, a corn soybean rotation in the Midwest, that would involve applying around 180 pounds of actual N. So there's quite a wide range, is my point here. The average might be, in this particular case, a 70 bushel per acre response. They've also acknowledged the fact that we tend to get a higher response to nitrogen at a higher plant density than we do at a more moderate plant density. And so in this research, they're often looking at, let's say, plant densities of 32,000 versus 38,000 plants per acre. These are the same results now shown for 2019, lower levels of response, and that's I think associated with the fact that 2019 was very cool and wet spring and we had delayed planting. So and now the response to nitrogen varies from about 25 to perhaps 105. And again, it's the same thing. It's a response to low versus high, where the low is at an average of application of only 60 pounds of nitrogen per acre. So there's a lot of hybrid variation. Now, if you were to choose hybrids, would you choose them for just how responsive they are to nitrogen? Would that affect your hybrid choice? Or would that be more likely to affect your nitrogen management in terms of what rate of nitrogen would you apply in order to achieve the optimum results for that hybrid? Well, that gets pretty complicated, and this is one of the tricky situations that I wanted to just allude to here, because I think it's fundamental when we look at results from commercial companies in the sense of, you know, suggesting a nitrogen responsive hybrid, let's say, versus non-responsive hybrid. And the way I'm going to do that is just to illustrate some results from our own research at Purdue, where we looked at 5 DeKalb hybrids in 2018, and we grew them at 4 different nitrogen rates that are shown here: 0, 75 pounds, 150 pounds, and 200 pounds. And so each color is a different hybrid in this particular case. Now, if you were to choose hybrid in this particular case, based on the yields at 200 pounds per acre, you would come to the conclusion that there's no yield difference in these hybrids, and you could pick any one of them and they'd be somewhere between 261 and 269 bushels per acre in this particular example. On the other hand, if you were to choose based on having the most nitrogen responsive hybrid, the highest degree of response to nitrogen, you would choose this green hybrid, which shows a gain of about 160 bushels to going from 0 to the 200 rate. On the other hand, if you would have more money in the pocket for a lower nitrogen application, if you were to choose the red hybrid here, because in this case, you can achieve 275 bushels with 50 less units of nitrogen. And once again, your nitrogen responsiveness is somewhere in the neighborhood of 148 bushels to that low versus a high response. And for that same level of nitrogen, you definitely would not want to have the green hybrid in this particular case, because it's yielding significantly lower than 3 of these hybrids. And so I want to acknowledge the fact that yes, hybrids are going to result in variation in the amount of nitrogen that's required to get to an optimum yield. And at the same time, I want to push us in the direction of trying to, moderate our nitrogen rates, or to change our nitrogen timing to better synchronize with plant nitrogen uptake, to limit our negative environmental impacts associated from applying too much nitrogen fertilizer, or the combination of too much manure plus nitrogen fertilizer. So when we get to the nitrogen fertilizer, I won't talk about these today, but basically we need to acknowledge the 4 principles, and a large part of this, of course, begins with the right rate. Okay, having said that, we also need to acknowledge the fact that in modern hybrids, it's quite common for modern hybrids to be taking up more of their total nitrogen in the post-flowering period. And this is illustrated in some work that one of my previous graduate students, Sarah Mueller, did, where basically she looked at old air hybrids. Those were the hybrids up to 1990, and new air, which were hybrids from 1990 onward. And she looked at the total amount of nitrogen that was accumulated up to R1, or up to the silking period. These were 4 studies which were at the same rate of total nitrogen, averaging 152 pounds of nitrogen. And these showed that 100 pounds was accumulated up to R1, and 110 pounds in the more modern hybrids. But the biggest difference between the modern hybrid and the older hybrids was in this post flowering period, in which case we got 64 versus 46 pounds of nitrogen. And so modern hybrids in this particular case were yielding more. They were yielding about 43 bushels per acre more. And some of that definitely is associated with the fact that we're picking up more nitrogen. We're probably staying green longer, and that means we've got more active photosynthesis that is meeting the needs of the grain kernels that are being formed. So how are these higher yields in modern hybrids being achieved? I've talked a little bit about the stress tolerant hybrids. I've talked a little bit about the timing of nutrient uptake changing, as well as the tolerance to progressively higher plant densities. Well, we know that in order to get to higher yields, it's tricky, but we know that we need high leaf area and functional photosynthesis. We have to be producing more photosynthesis than we're respiring away during the dark nights, during the grain filling period. Number 2, we want to have low stress at flowering. We want obviously to have good synchrony in silk emergence and pollination, and limited abortion afterwards. We want to have ideally cool temperatures that extend the grain filling period duration. And some of our higher yields in Indiana this past year were associated with just having cooler nights in the grain filling period, as well as higher levels of incoming solar radiation, because we tended to be somewhat on the dry side and had less clouds than we did in most previous years during the grain filling period. And then we need a strong ear sink demand, because what we'd like to do in order to get to higher yields is to have around 60% of the total plant weight at maturity be in the kernels. And of course, we can't have weak stalks in that process. We need to have strong stalks to limit lodging. But what's less understood for specific hybrids in each production environment is everything to do with how much do we need to optimize kernel number versus kernel weight. And so in today's presentation, I'm going to talk a little bit more thoroughly about the kernel number versus kernel weight question. And getting to bigger corn, I'm going to argue today is perhaps more about, in today's hybrids, is more about getting higher kernel weights than it is about getting more and more kernels per plant or more and more kernels per unit area. And so when you think of kernel number is by and large a function of fairly early in the vegetative period. It's a function of the number of rows being determined right around V7 or something in that neighborhood, a number of rows of kernels, and then kernels per row is being determined up to about V12. And then you could say, okay, yes, it's true that during the post-flowering period, that there could be some abortion of kernels occurring. But when you get to kernel weight is by and large a function of just the reproductive period itself. And what happens in the reproductive period itself is that these kernels are for the first 2 weeks, for the first 15 days or so on, are really not gaining any weight after they are pollinated. What's happening in this period of time is that there's just a very rapid cell division going on, but they're not accumulating very much weight for the first 2 weeks, and we call this the lag period. So there's really almost no weight gain during this time period. And then we go to what's called the linear period of grain fill. And we want this period to be as steep as possible and as long as possible to get to the highest individual kernel weights possible. And so this is part of our management strategy. Our management strategy is not just to get to a high kernel number, but is to create an optimum situation in the plant to try to get to higher individual kernel weights by having a steeper and longer grain filling period. And longer is helped somewhat by having cooler nights during the grain filling period, but it's helped even more by having healthier plants during that time in terms of low disease incidence and in terms of adequate nutrition. So we need to consider then just sort of whether or not that's true and how that changes with hybrids and with plant density. So one of the illustrations here is from DeKalb hybrids that we grew in a 38-year period from 1967 to 2005. We grew those at a couple of locations at 2 different nitrogen treatments and at 3 plant densities that I'll get to in a minute here. So these hybrids from the different decades were grown side by side, and these are the yields, first of all, at 200 pounds of nitrogen, and then these are the yields here at 50 pounds of nitrogen. And so what we're getting here is an increase of about 1.6 bushels per acre per year at the higher nitrogen rate, which kind of is close to the rate in average for the U.S. During that time period. That result was averaged over the 3 plant densities. We grew these at 3 different plant densities, 22,000, 32,000, and 42,000. 42,000 is is shown here in the brown. And so here are the yields for the 42,000 shown in this highlighted brown line here, whereas the black is showing the yield results with 32,000. So obviously we're getting highest yields with the 32,000. We're not gaining with the 42,000. And we're in this particular case, we're just for one of the two locations, we're averaging 1.5 bushels per acre per year. Where did that yield come from? Did that yield come from higher kernel number, or did that yield come from higher kernel weight? Well, we looked at all of those questions during this study. And as you would expect, we had lower kernel number for 22,000 in the blue than we did for 32,000 here in the black. But what struck me the most is that this is about 5,000 per meter squared. So you have to think in terms of something in the neighborhood of 4,500 per square yard of kernels in this situation. And so if you had 32,000, you probably have about 8 plants per square meter. And so this is averaging somewhere around 650 kernels per plant when we get to these more modern hybrid situations. But the biggest response in terms of getting to higher yields was with kernel weights during this time period. And so this began me down the road of trying to look more intensively at the reproductive period and meeting the corn plant nutrient demands in the formation of kernel weight. Another study that kind of pushed me in the direction of looking more at individual kernel weights rather than number is a pioneer study. In this case, we grew hybrids side by side over representing 70 years of improvement. These hybrids were first commercialized in 1946 through to 2015. All of these hybrids are perhaps late for what your normal production system would be. These are averaging about 113 days in maturity. And these were grown at 5 different nitrogen managements. And for today, we're just going to concentrate on the difference between the 0 and the 200, where 200 pounds of nitrogen was applied with a UAN between the rows at the B3 to B4 period. So that's one we're going to focus on. And again, these are hybrids grown at the same plant density. So all of them are grown at 32,000 plants per acre. And as I showed earlier, the 2015 hybrid is going to be staying green longer than the 1958 hybrid. And these were the yield results. In this case, we're gaining over this time period, 70-year time period, we're gaining 2 bushels per acre per year. But we're still, even if we don't apply any nitrogen, we're still gaining about a bushel or 0.8 of a bushel per acre per year. So did that come from kernel number or kernel weight? Well, these are the kernel numbers. In this case, I'm expressing them on a per plant basis because we only had this study at 1 plant density, and every hybrid had achieved the same final plant density. And you see that this particular case, we got to about just over 500, but there was no increase after 1976. On the other hand, when it came to kernel weights, the kernel weights, individual kernel weights, and this is expressed as milligrams per kernel, we're going from about 230, let's say, up to over 300 with this higher nitrogen. So we're pretty sure that the changes, especially in the last 20 years in corn hybrids, has been a realization that we can only push plant density so far, and then because if we push them too far, we might pay for it, especially in not realizing the kernel weights that we need to get to in order to have a corn plant at the end of the day that has a large 60% or so of its weight in the grain, especially for a grain production scenario. We recognize that in that scenario, though, there is lower grain end concentrations. Hopefully.
All right, buddy, you're back. Yeah, we lost you for a little bit.
I'm sorry about that.
No, that's what we worried about happening.
I'm at the university, so let's see. Do you still see my screen, Mike, or do I need to?
I do not see your screen. So you need to share screen again.
Let me go back to it then. Hopefully this will work, and my apologies. I'm at the university, but.
We know that means nothing anymore. It doesn't matter where you're at.
Okay. So anyway, this is we know that we are losing nitrogen concentration in the grain.
We still can't see your screen, Tony.
Still can't see the screen.
No.
It says I am screen share.
And we lost him again. Yeah, for half an hour. Yeah, Tony, we lost you again, buddy. You froze up. And you're back.
And back again. Let's try that again.
Let's try to see if that screen can get up.
I hope the screen can return.
There it is. You're good.
It is? Oh, yep. Thankfully.
Let's hope we can keep it going for another half hour.
All right. So this is the grain nitrogen concentrations, and they're declining in these modern hybrids. Now, that's not always the case because sometimes the modern corn hybrids are also quite good at increasing the grain nitrogen concentration when higher rates of N fertilizer are applied. And so one of the ways in which modern farmers capture nitrogen that they apply to their fields is not just in realizing higher yields, but in realizing an increase in the protein level of the grain in the finished product. So in this, I'm going to go on to just in the interest of time, I'm going to, so I'm going to go on to here to basically say that one of the ways in which yields are increasing, we typically think in terms of meeting nutrient needs of corn plants during vegetative growth. And I'm going to perhaps point you in a little bit of a different direction, which is basically to say, our nutrient levels are perhaps even more important during the grain filling period than they are during the vegetative period in terms of realizing higher grain yields. And that's shown a little bit in this graph, where essentially you're looking at the nitrogen concentration of the leaves at maturity, and basically the nitrogen concentration in the leaves are shown at the bottom here. I'm trying to find my mouse, and I'm not sure I'm going to find it somewhere in here. Perhaps it'll show up. Anyway, the 2015 results basically show higher nitrogen concentrations in the leaves at maturity going up to 1% or 1.2% resulted in 300 bushel per acre yields. And so it takes not just adequate nutrition during the vegetative period, it takes adequate nutrition that extends itself through the grain filling period. And the same thing is true for a nutrient like potassium. So here again is the same hybrid scenario from the older hybrids to the newer hybrids. And in this case, it shows leaf potassium levels at maturity. And so I guess I want to make the argument here that that we need to be thinking in terms of adequate plant nutrition to keep the plant healthy all the way through grain fill in order to realize the higher kernel weights that I believe are necessary in modern systems. So why is that? Well, part of it is the fact that that in these modern hybrids we are in effect realizing a higher harvest index. So in other words, more of the grain is dominating the weight of the final plant. And so now we're often getting to 59, 60% of the weight is in the grain. And that also means because we have more weight in the grain, and we can also get more nitrogen removed from the field, as well as more potassium removed from the field in the grain itself, because we're achieving the higher grain harvest index. It's fair to point out, though, that not everything is rosy if you come into a drought stress. And so this is an illustration here of what happens to kernel weights, expressed here as a fraction of a gram. So in other words, 0.3 here would be the same thing as 300 milligrams. But basically, it shows that basically if you have a drought scenario shown by that lower gray line, then you can essentially have almost no gain in kernel weights over the whole time period. So of these hybrids from different eras. So in going then to timing of nitrogen as perhaps one strategy to keep the plant healthier longer during the grain filling period, I want to just sort of illustrate, the range of pre-plant, at-plant, side dress, progressively later side dress applications. And later timing isn't always going to give a yield increase. And I don't want to pretend that every producer has to go this way. But I do want to talk through some of the scenarios that occur and that either lead to a good scenario where positive yields are obtained with a later timing, and in some cases, no gain is made with a later timing than, let's say, the traditional side dress of around the V6 stage. So in our case, we applied using Y drops in a 3-year study, and we applied our results here, shown here. I'm trying to find my mouse and I don't know where it's ending up, but hopefully I can get it back again somehow. So we found that there were yield increases associated with going, let's say, from 0 to 250 pounds of nitrogen. And these letters above kind of show whether or not there was a significant difference when we weighted in the gray bars to applying the last 50 pounds at the B12 stage. So what that means, let's say, for the 150 pound rate is that it was 150 pounds in the blue applied at planting. In the green, it was 75 at planting, 75 at B6, and in the gray, it was essentially having 100 pounds at planting, and then the last 50 pounds were applied at the B12 stage for that nitrogen rate. That was 2016, and here are the results from 2017. And now there is an increase with the split application, although there is no difference for the B6 versus the B12. And in actual fact, if you go right up to the 250 pound rate, there was a bit of a yield reduction for the B12 late application. But what was interesting to me, again, coming back to this scenario of the overall relationships here, is that when we got to these higher grain yield levels, it was almost always because we achieved higher kernel weights. And so that's shown here in this graph. And how did that come about? Well, earlier I referred to essentially having a lag period, and then showing basically what happens in the linear period. And so what is the difference in achieving, let's say, 231 milligram kernel weights in the left side? In the middle, we see we got to 299 kernel weights, and in the right-hand side, we got to 321 milligram kernel weights. And that allowed us to achieve an overall yield of 254 bushels. Well, the difference is basically is how long is that linear period. And so we got to the higher yields on the right-hand side because instead of having a linear period of 51 days like we had on the left-hand side, on the right-hand side, all of our filling period was continuing for another day or two. And so higher yields with using the route of higher kernel weights are usually achieved by having a longer grain filling period effectively. So is this harder to do at higher plant densities is often the question we have raised. And I will acknowledge that's a case. And so this was some grain yields from different plant densities, again, 22K, 32K, 42K, and showing the response to 0, 100, and 200 pounds of nitrogen. And the highest yield here was in the middle there at 32K at 200 pounds of nitrogen. The nitrogen in these trials was about 70% of the nitrogen was in the plant at where the arrow is here in the middle at the time of the R1 stage. So basically during the vegetative growth, 70% of the nitrogen was accumulated and another 30% of the nitrogen was added after that time period. And so what we're doing when we try to apply some of our nitrogen later is try to meet the nitrogen demand during that grain filling period. And this essentially shows what the responses are in terms of other nutrients that are going to be coming into the corn plant. I've retrieved my mouse again. I don't know where it was all of this time. Sorry about that. But anyway, so this is the same nitrogen figure here, which is basically 70% in at the R1 stage. Other nutrients that are also of interest are sulfur. There's been a lot of positive responses of corn to ammonium sulfate or ammonium thiosulfate. And basically that's not just a vegetative stage phenomenon because of the fact that the sulfur is about 42% of the sulfur is going in after the R1 stage. Once again, with phosphorus, about 55% of the phosphorus is going into the corn plant after the R1 period. And about 55% of the zinc is going in afterwards. And so we've got a lot of nutrients that are going into the corn plants, representing new uptake during the grain filling period. Now, when it comes to plant tissue testing, the most common route for plant tissue testing has traditionally been to look at the sufficiency achieved at the R1 time period. And this has effectively been shown here as what do you need in terms of nitrogen in Indiana, and for our Ohio, Michigan, Indiana area, we recommend 2.9% nitrogen in the year leaves at the R1 stage. And this is a graph showing a lot of experiments over the years from 2010 to 2016, showing what kind of yields we got with what kind of ear leaf nitrogen concentrations. And you can certainly see that that perhaps 2.8, in other words, 28 or 2.8% represents a cutoff that is very important to reach. The trouble with analyzing at the R1 stage is that basically it doesn't tell you how long the leaves are going to stay green during the grain filling period. And it may not be predictive of what's going on in the rest of the canopy, because usually in corn plants, the leaves that are the last to die off, the last to turn yellow, are those right in the middle where the ear leaf is positioned. So getting high nutrient concentrations at R1 stage is more difficult to do when you have higher plant densities. And so here's an example of 22,000, 32,000, and 42,000, and what the ear leaf nitrogen concentrations were when we applied 200 pounds of nitrogen. And these are shown here at 2.6, 2.5, and 2.4. And so I do want to make the point that, and this is phosphorus concentrations here. So I do want to make the point that when it comes to zinc, phosphorus, and nitrogen, it is harder to get to critical concentrations when we have higher plant densities. And so one of the reasons why higher plant densities aren't necessarily always positive is the fact that maintaining adequate nutrition for every plant is harder to achieve. And so this is also shown here for ear leaf phosphorus concentrations and ear leaf potassium concentrations. We usually recommend 19 grams per kilogram, which is the same thing as 1.9%. And you can see that we have a lot of yield gain that still occurs to 260 or more when we have higher potassium levels. And over the last 5 years of research, I've tended to find that the 1.9% is not enough for potassium, and that we really ought to be at about 2.1 to 2.3% potassium in order to get to higher yield levels. And with sulfur, we'd ideally like to be around 0.2%. And then the other part of getting to better plant nutrition is not just looking at an individual nutrient, but looking at the combined impacts of nutrients. Now, perhaps where you're in a dairy farm situation, you're returning a lot of dairy manure, you're adequately addressing the potassium needs of your soils. In much of the Midwest that relies on cash crop production, potassium levels in the soil are too low for optimum yields. And that is not new information. That was known, 22 years ago, I guess, or 23 years ago. And this is in an Illinois study where they looked at the response to nitrogen going from 0 to 240 pounds. And this white line here is the response to nitrogen, where they also added 144 pounds of K2O. And so what's illustrated here is the very small response if you didn't add potassium at the same time to that situation. And the same thing is true in more recent research from Antonio Mallarino in Iowa State, basically, and this unfortunately is in metric units. So this would be about 200 bushel corn right here. And we gain 20 to 30 bushels per acre at these higher nitrogen rates of, let's say, 230 pounds per acre when we have added potassium. And if we don't add potassium, we get a plateau in the nitrogen response much sooner. And the same thing is true here. In this case, the borderline yield here would be about 210 bushels per acre versus 240 bushels per acre when we added potassium. So response to nitrogen may sometimes be limited by insufficient potassium in the corn plants, and that's harder to achieve when you have progressively higher plant densities. And then there's a whole host of other nutrients that I simply won't have time to talk about today, except to acknowledge just the high amount that is going into the corn plant during the grain filling period, and that is needed to meet the kernel requirements of these nutrients. So to wrap up and to allow time for questions, I'm going to basically just say it's tricky. It is difficult. And it takes good management and high prices or higher prices in order to survive in corn production for yourself and for the next generation of farmers that will follow you. So is it easy to achieve bigger corn with today's hybrids? Well, it's still very difficult. Yes, it's true that modern corn hybrids require less nitrogen per unit of yield, but I believe that to get there, we need to have more balanced nutrition, especially available around the flowering time during that lag phase, when the kernels are setting up to figure out how many cells are they going to divide, what is the potential
Tony, you froze up again, buddy. We're so close, but you are froze up. Well, hopefully he can get back on like he did last time, because we were so close. Again, if you guys have questions, type them in that Q&A box there. We will have, if he gets back on here, we could have a couple minutes for questions. Hopefully we get him back. If you have questions, you can type them anyway. If we don't get them back, I can email Tony the questions, and hopefully we can get him to email a response to you if you have a specific question about today's presentation. Well, I see some questions, guys. I appreciate that, but I'm not sure we're going to get him back here. Tell you what, let's take our five. Well, let me see. I see him. There he is. He's popping back up. We were so close, Tony.
Yeah, we were very close. I'm sorry, but I'm going to try to share the screen. If I could be enabled to share the screen again, I'll just finish up here.
Okay. Brandy? I think she's working on it.
My apologies for the poor connection from Purdue.
Well, I think it's what we're going to have to deal with here in all of our meetings this winter. Bernie, are you able to get that back to him?
Yes, I'm.
There you go. You're good.
Perhaps getting closer now.
Yep, you're there. I see you.
You see it? Okay. So
Yep.
basically, point number 2 under tricky is what changes in management? Well, I think they begin with spending more time in the field because I believe it's very important to be thinking in terms of no plant left behind. In other words, as we go to progressively higher plant densities, ensure that all plants are responding uniformly within the row so that the competition is even. And I believe that we need to be more focused on plant tissue sampling at earlier stages as well as later stages during the grain filling period in order to make sure that we're meeting the needs of corn plants. So these 3 suggestions I would have to tailor nutrient management to specific hybrids. First of all, if you're going to go in this direction, and I think it's a good thing that seed companies try to learn more about the agronomy of the hybrids that they sell. But I think if you want to go that way specifically, you want to have as much background information about yield responses to specific nutrients across range of rates, timings, and plant populations. If you want to adapt your management to specific hybrids, I think we need to know more about the relative stay green with or without fungicide application and its flexibility, both in terms of kernel number and kernel weight, that point to the likelihood of a response to later season nutrient application, whether that later season nutrient is nitrogen or something else. And then we can't go down the route of overemphasizing any one nutrient, and nutrient balance is extremely important. And in our world here, a lot of that is associated with getting to a better balance between nutrient of nitrogen and potassium to try to keep those two in about a 1-to-1 ratio during the lifespan of the corn plant. So there's other factors that I didn't talk about here, and I want to acknowledge the graduate students and the support of Indiana Corn Marketing Council. Farmers are very important in terms of the applied research that they support, and the companies have also provided support for individual graduate student projects over the years. And I want to close by apologizing for all of the interruptions and to suggest that I'll be happy to answer questions. So thank you, and appreciate your patience today.
Tony, thank you, buddy. I mean, this is reality, and I tell you, I've had worse. So we got you back every time, and that's a bonus. But hey, great job. There are quite a few questions. I don't know. Can you open the Q&A, or do you want me to read them to you?
Let me see if I can get to Q&A. Yes, I can get to Q&A. How was the potassium applied in the last study? It was broadcast applied prior to planting and incorporated before planting. Jeff Miller asks, is it correct from your graphs that a split application of nitrogen and at planting and at B6 is the most economical? and that sufficient sulfur. Yes, we're in the did with that hybrid combination. We found that there was no advantage from a B12 application when it came to yield. But sometimes there can be an advantage from a B12 application when it comes to being able to lower the overall nitrogen rate applied and get the same yield. And so, and because typically what happens with a B12 application is that we get a higher nitrogen fertilizer recovery efficiency. More of the nitrogen we applied ends up in the corn plants. Sufficient sulfur and potassium are very important. Yes, we're typically getting around 20 to 30 bushel yield responses here in corn to making sure that we've got adequate sulfur and potassium. And a large part of that is associated with keeping leaf potassium levels higher during the grain filling period. Have we looked at foliar applied fungicides at the R1 stage, and how has that impacted the linear period? I have not, but yeah, to the extent that the fungicide is preventing a higher level of disease incidence and keeping the canopy green longer, it would work well towards doing one of two things, either making the linear period longer or enabling a higher rate of photosynthesis per day so that you can increase the steepness of that linear period. Was there another question here?
Yeah.
Okay, higher plant density require more water to get enough nutrient in to maximize yields. Okay, so this is John or Jody, I guess it is. Or no, it's from Ron.
Ron Grusha.
Right, I'm sorry. It's from Ron. Do you need more water? That's a hard question to answer. One of the things that limits us most with late season surface applied nitrogen is having rainfall, a half inch of rainfall occur within, let's say, 5 days of application. If we're applying at the B12 stage and we don't get rainfall for 2 weeks and we've applied it on the surface, that obviously isn't going to help us. To some extent, more water is good, and I'm kind of obviously do not have enough in the way of side-by-side comparisons at different watering levels to be able to answer that question entirely. But I do believe that corn plants are pretty resilient today, as long as they have good depth of rooting and as long as the soil has not been compacted and if you have good soil structure. And so it surprises me sometimes how well modern corn plants can do despite having, you know, a 3 or 4 week period with very little rainfall. Another question is
just above there, you missed it, Tony. There's 2 above there. David DeGore asked, any response to boron and N application with Y drop?
We have observed responses to boron application, although usually what we've done on the boron application is apply the Aspire combination. So that's adding, happens to be a potash fertilizer source that has 58% K2O and then half of 1% boron, and the boron that's in there is is got two different formulations of boron. I prefer to have boron available earlier than, let's say, a late Y drop application. The critical levels of boron are an area that we don't know enough about. And we usually say we want to have around 4 parts per million in the ear leaves at around the flowering time in order to have sufficient boron. And my guess is that that level is not enough to achieve the best possible growth of the silk channels and the best possible pollination establishment. So with Y drops, my experience on those has been only the UAN application. I've not added boron that way. Most of my boron work has been with either before planting or early application up to B6 stage with in-season Aspire application. Was there something else I'm missing?
Yeah, so right underneath that question, there's one from Eric Zuber about, he says, does kernel weight have an effect on drying time and drying cost at equal moisture?
Pretty minimal.
Okay.
So it would have almost zero impact. I think what's going to affect drying costs, well, I mean, obviously, if we're going after higher kernel weights and we've got a slightly longer grain filling period, then we're going to have somewhat higher grain moisture contents. But that's a pretty minimal response. It wouldn't make more than a 1 percentage point difference if you're harvesting in the range of 24%. And it would have almost zero impact on drying energy.
Okay. There's one more down below I think we can cover before, as our time's getting away, but Martin Willis says, have you ever done any research on kernel number versus kernel weight in silage corn yields and quality? Oh, did we lose him? Yeah, he froze again. Well, folks, that might be it for Tony. We got a little bit over time, but there's a lot of questions about his presentation. So I will ask Tony, you guys will all have a recording of this that you'll have access to, for the last 2 days. But I will also ask Tony, because a lot of people have asked for his presentation. And if he allows that, then I will get that out to those folks. Oh, he's back. Can you hear us?
Yes, I can hear you. And
okay, yeah.
apologies again.
No, it's okay. I think we're at this point where our time is going. There was one question about corn silage that I was talking about.
Yeah. So as you know, I mean, a large part of the grain component in corn silage is the fact that it's needed to dry corn plants down quickly so that they get to the safe silage storage situation. But it's of interest to me that in silage corn, we're going in the direction of plants that have progressively higher harvest index and progressively more of the nitrogen is in the grain. And the biggest change that's occurring is less nitrogen in the stem. And so hybrids are going in the direction of having less stem nitrogen and a bigger fraction of the nitrogen going into the grain when they're meeting the demands of these bigger kernels. So I haven't done any research on whole plant corn silage, and I realize how important it is in your state. You guys really need to hire a replacement for Bill Cox.
Amen. No. Yes, we have a couple positions that now have been vacant for over 3 years. And we do not have a state agronomist.
That's sad.
Yes. Well, Tony, that was great. There is one other question. I don't know if you can stick around and answer that. Actually, you know, if you can type your answer into that box.
I'll answer live. Yeah, please get on with your program, and my apologies
Yeah.
for all these interruptions.
No, and I tell you, Tony, I'm getting a lot of text things that people are asking if they can have a copy of your slide. They want some of these slides they really want. So I don't know if you you do that, provide a copy of your presentation for folks in a protected view, or if there's, I mean, they'll have it viewed by video, but a lot of people are asking, I want that certain slide. I want this slide.
Oh my goodness. Yeah, I'll be able to provide it first of all to you, and then we'll figure out a way to answer these questions. And
Okay.
thank you again.
Fantastic, Tony. Again, appreciate your time.
Okay.
Have a great rest of your day. Appreciate it.
Bye.
All right, folks. I'm going to give you a 5-minute break here just to stand up, hit the bathroom, get a drink, close your eyes. Would like to play the slides again for our sponsors here. They're very important for us, you know, to keep us going here with the shortened 2-day Congress into a 4-hour Congress and things. So we do appreciate our sponsorships today. So we're going to put those up. And it's 10 after now. So I'm gonna 15 after. If you guys come back at quarter after, and we'll start with Ken Wise from the New York State IPM program, talking about some of our black cutworm, corn, western bean cutworm, and armyworm population. So I'll see you in 5 minutes. All right. All right, everybody, it is quarter after. I want to keep us moving. We're going to continue. I see Tony's still, he must be still answering questions. So thank you, Tony. All right, Ken is sharing his screen. So Ken Wise is kind of our counterpart here in the New York State IPM program with field crops and livestock. Ken's been around as long as I am. He's a 20-year pluser out there from the east side of New York, covers the whole state. But he's kind of been in charge, you know, since we started doing some of this pheromone trapping. Ken is in charge of kind of getting all that data together from all of us here in the regions, and he manages all that. You know, some of the good charts and things that I show you usually at Corn Congress, they're from Ken, so they're not from me. So I've asked Ken, you know, to, instead of me doing some of this, to say, Ken, would you like to come on and talk about some of this information you've put together on these three, you know, the three insects we monitor for, being the black cutworm, the true armyworm, and the western bean cutworm. So Ken is here today to talk a little bit about that and the importance of some of this pheromone trapping. So Ken, you're good to go.
All right, thank you, Mike. So I'm gonna talk again, like Mike said, about some pheromone trapping that we've done over the years. Most of our pheromone trapping has been about western bean cutworm, but in the last few years, we've been doing more and more of the black cutworm and true armyworm trapping. Let's see if I can get this to work. There we go. This, so our funding for this last year came from the New York Corn and Soybean Growers Association, and you know, we're greatly appreciative of that funding, because it's, you know, it's getting harder and harder to find funding that you can do this kind of project. And so what I'll do is I'll go through some of the biologies of the three pests that we're talking about, and then the data that we collected in some cases since 2010 to current, and then for black cutworm and true armyworm will be the last 2 years. So the purpose of the pheromone trapping network for field corn is to, for the most part, to give an early alert system to high level of flights of these moths that occur. So the black cutworm and the true armyworm, they occur early in the season, and the western bean cutworm occurs later in the season. And there are slightly different purposes of monitoring the flights of the two early ones versus the western bean cutworm, which I'll get into later. And you know, it's a way to identify different areas of the state that, you know, within a season is a good area to go scout. So say your farm was next to one of these traps, then, you know, you get a high population. Doesn't mean you're going to have a problem, but it could mean that you might have a higher risk of that pest being in those fields. So these are the three species that we're monitoring for now. Western bean cutworm was found in New York in 2019. And so we've been monitoring that one for quite a while now. Black cutworm and true armyworm are more recent. We started 3 years ago, and we've only collected the data the last 2 years. So, you know, that I wish I would have had a third year to show you, but there was some problems with collecting it and we lost some of the data. But, you know, we got pretty firm data the last couple of years. And what we do is we put these pheromone traps in and out next to fields, cornfields, and there's a little pheromone goes in a little trap in the top that the male moth is attracted to from these different species. And you put a specific pheromone for the specific pest. So if you have two different pests that you're monitoring, say you do monitor black cutworm and true armyworm at the same time, you have to use two separate traps. So those go in next to the field. There's a insecticide strip inside the bucket. So as they fly in there, they get hit with that insecticide and then they go down into the bucket, and we can count on a weekly basis what we're catching in those different traps. So again, you know, we started in April, and I know Mike Stanyard last year started in early April collecting, and he did find some black cutworm and I believe possibly true armyworm relatively early in April. So they do come in early on storms, and they are looking to lay their eggs on grassy weeds and that type of thing. Maybe not so much specific right on the corn, but I'll kind of give you an idea why that would be. And then late June, and then from late June to late August, we start monitoring for Western bean cutworm. So, many of you already know this, maybe even better than I do. Black cutworm is a larvae of a moth. It is in a field, it is a greasy pale gray to blackish in color. They have black spots on each segment. They range from about 1/8 of an inch to 2 inches. And they have 6 larval instars. So they range in different sizes over time. And this is what they do. They will cut your plant off early in the season. Cutting the plants off, you're eliminating those plants from being producing any kind of grain or silage. This is their life cycle. And so they migrate from the south or out towards the southern Midwest up into the Northeast. I think we get on the east side of the state, we get our populations coming up the coastline sometimes. But I know that like Western New York, where you guys are, I think you get more of your populations coming in up the Ohio River Valley, those range, those storms and weather fronts. They will come into a field, they will come up on storms, they will lay eggs, generally again, maybe earlier than you have your corn planted on grasses. So what happens is that you'll go in, you'll spray out your weeds, you plant your corn, and then those larvae hatch and don't have anything else to feed on. So they're going to be feeding on your corn and just cutting it off like that. So once they fed on your corn, they get to that 6th larval instar, they will pupate. And they pupate in the soil. And you can get a 2nd generation black cutworm. Generally, your corn has gotten too large for them to chew on at that point. So at least with black cutworm, you don't generally have an issue with the 2nd generation. If we have some late planted corn, I guess you might get an issue with that, but generally it's just that 1st generation that's here. And the economic threshold that we use that's in the Cornell guide that Ellison Shields puts together is suggesting that if 5% of the plants in a field are cut, like as you see in the picture, they can also they also feed up onto the stem a little bit higher and can notch them out, but rendering them wilted and eventually they die. If you're getting 5%, then you probably need to either spray the area that they're in or if it's widespread, then you may have to spray a whole field. So this is what you're going to try to avoid. And I'll get into some management issues towards the end of the biology part of this. So our second pest is true armyworm. And true armyworm, just like black cutworm, flies on storms. They lay their eggs in on grasses. They kind of probably prefer hay fields and the sort also, but they will lay their eggs on grassy weeds in a cornfield. So if you have like a no-till field that has a lot of grassy weeds early, again, if you spray them out, those armyworms will be switching over to those corn plants. Some of the issues with armyworm and possibly black cutworm is that you might get multiple storms, say from 1 week to the next, or a couple of weeks maybe, and these will lay their eggs at different times. So in the picture here that Keith Waldron, had taken years ago, you can see that you can get a mixed sizes of larvae, and that's a little bit harder to make economic decisions on whether to treat or not. But if they come in, they lay their eggs, and they all feed at the same time and grow at the same rate, it's a little bit easier to make some of those economic decisions, threshold decisions on true armyworm relative to corn. So this is what they look like. This is a mature larvae. They do have kind of variations of different colors. This is a general description of it. They say they have kind of an orange stripes going down either side. To me, they kind of look more of a yellowish kind of orangish color, and depending on the actual instar that they're in, the molt that they're in. And they also have a white stripe going down their back. One of the giveaways for armyworm, if you're out searching, is that they do feed from the edge of the leaf into the midrib, and they leave this frass, as you can see here, up on the plant. So to me, if it's that time of the year where you're your corn's in whirl stage and you're having this up on the plant, you got a good chance that that's a true armyworm. I'd like to point out there's a lot of different armyworms out there. This was originally called true armyworm. It has recently been changed to armyworm. I kind of keep it as true armyworm because it kind of depicts this species of Lepidopteran that attacks your corn, as compared to dingy armyworm or several others that we do see can have it out in fields. So this is the damage that they start with, and this is just some corn that I had monitored several years ago, and I'd found the armyworms, and this was in the no-till field, not a no-till field. This was a cover crop that had been broken down, and you had some armyworm damage, this is pretty minor, but this is what they do. They feed from the edge into the midrib. Let me go back to that quick. And if the plants are larger and you have a high population of these insects in a field, they will feed on the leaf area and will leave the midrib. And I don't have a picture of it, but they can look like pineapple fields, kind of like with a bunch of just stringy midribs sticking out of the corn. They may recover. You're probably going to have some yield loss if that's the case. But the difference between this one and maybe black cutworm is that armyworm can occur at extreme numbers. So they're both nocturnal, meaning they both feed at night for the most part. But when armyworm is in extreme populations, they can march during the day, and they will go into a field and will feed, and then we'll keep going and go to the next field. And the thing about both these insects is that they have 6 larval instars. In that last larval instar, they eat 80% of the forage that they're going to feed on, which means, relative to armyworm, and I've heard growers tell me this, like yesterday the field was fine and today it's just gone, and that's only because you hadn't noticed them feeding before because maybe the damage was rather minor, but you had a high population of these things and they reached that final instar at the same time, and they ate a lot of forage, a day or two. This is the life cycle of true armyworm. Again, they migrate from the south, just like black cutworm. They lay their eggs on kind of grassy weeds or hay fields. Once you spray out those weeds, they do feed on the corn, and again, they feed from the edge of the leaf into the midrib. There is a difference here. I mean, they do pupate in the soil like black cutworm, but they also, we didn't think that the second generation really caused us an issue. But a few years ago, we had a second generation of true armyworm that was causing issues with corn and sorghum Sudan grass. And this last summer, we also had, I don't know if it was a true second generation or if we might have had another late migration of moths that come in on storms in the summer, but they really nailed some sorghum Sudan grass fields across northern New York and out in western New York. So generally, we only monitor this insect pest up until maybe mid-June at the latest. I think for armyworm specifically, we're going to try to monitor that possible second generation, into July to see if we're getting flights of those moths later on in the summer. And that could be an indication of a second generation, or like I said, it could be just another flight of them coming in on storms. So our economic threshold for this pest on corn, and this is straight out of the Cornell guide, is that if most of the plants are showing damage and you're having 3 or more larvae per plant and they're still small, and there's no biological control agents on them, meaning parasitoids or that or what have you, you know, it's a good idea to go out there and spray those corn plants or those fields. The point with the biological control, though, it was interesting last summer when those sorghum Sudan grass fields, that almost every picture people sent me from those fields had what we call parasitoid eggs on them. So little tiny white eggs that were laid by parasitoids. Once they hatch, and they will feed and destroy those armyworms. So it would have been interesting to see how soon those would have occurred if they hadn't sprayed them out. But yeah, there are a lot of biological control agents that do, especially with armyworm, can help control them. It could be a disease, it could be another insect, tiny little wasp that lay their eggs in or on the larvae. There's also diseases, and you know, back in, I think it was 2001 or 2, we had a huge population of true armyworm. And you know, in some of the fields I was in, small grain fields, that the larvae climbed up the plant. It looked like they were going to feed on the grain, and they all died. So they had all got a disease at the same time and just wiped out the population. And occasionally you can have help from birds. Birds sometimes will come into a field and we'll feed them and can make a major effort at getting rid of those things, but you never know when and where they occur. So managing these pests, the best way to do it, at least with the BT trait, selecting a variety that is resistant, is a good way to go for managing black cutworm and true armyworm. And if you go to what they call the handy BT trait table, and this will be updated soon, I would guess a 2021 version will be coming out. It lists the hybrids and what insect pests are resistant to. And both black cutworm and true armyworm are on some of those varieties in this list. You know, if you're growing corn, this would be a good thing to have, even to print off and maybe put on your desk, just so that you can kind of get an idea of, and you know, they list out the silage varieties versus the grain varieties and the sort. So the other way to control black cutworm, and you know, this is pretty much on all corn nowadays, at least into the near future, if we don't lose our neonic seed treatments, is the use of poncho or cruiser on your corn. These do, on the label it has black cutworm listed. It does not have true armyworm listed, at least what I didn't see yesterday. I didn't see it on there. But they do control black cutworm early in the season. So if you have your BT and your seed treatment, you're probably good to go for control of those two insect pests. I'd like to point out also, though, that the new Cornell guide for integrated field crop management, we worked with Elson this last year and putting together a new table of all the registered insecticides that you can use on corn that had research behind them relative to the pests that we're looking at. And you know, there's a nice table there that and a lot more insecticides that had not been in the guide in the past that you can go and use. And so hopefully that'll help you. But you know, if you need a product and you need to go in and spray them out, there are quite a list of them that you can use for those pests. So the way that works is that, you know, these are by crop. So this is by corn, and there's actually several other species of insects listed on this, but true armyworm and black cutworm were two of those that are. And then on the left here, you got all the different products that you can use in and on corn. But again, you know, you only want to use it when you reach an economic threshold and not really, you know, don't do it as an insurance spray. Western bean cutworm. So this is a completely different scenario. Those other insect pests that I talked about do not overwinter in New York. They have to come in on storms. You know, in 2009, we found Western bean cutworm in New York State. And it does stay here, it does overwinter, and it has built up over time. And so I'll kind of go through this. This is what the moth looks like. you know, it's good giveaway is it's got this white band along the side, kind of this crescent moon mark here and a dot, white dot here, if you happen to be monitoring for them in your own fields. And what they do is they overwinter in the soil in a pre-pupal case, and in the spring they pupate, and then they will emerge just before the corn tassels. So they want to they want to lay their eggs, and they are pretty specific. They will lay eggs on pre-tassel corn and generally not anything else. They will travel long distances in an effort to find pre-tassel corn. If it's tasseled or if it's pollinating already, generally, from at least from the literature that I've read, they do not lay their eggs on those kind of plants. Now, I have talked to some entomologists out from Indiana and out through the Midwest, and they do say when there are conditions are not good and that they cannot find a pre-tassel corn, they may dump their eggs onto some non-pre-tassel corn, but for the most part, that's what they're looking for. This is their life cycle. They overwinter as a pre-pupa, and then in the spring they pupate, and then they come out kind of pre-tassel. The eggs emerge, or sorry, the adults emerge pre-tassel. They will lay their eggs on the plant as seen in this picture, and they will develop over time. The eggs will turn purple, and these tiny little larvae will come out. And this is what you want to be scouting for. When you're out there looking, having your scouts, if you do it yourself or you have consultants doing it, you'll want to be monitoring these at the pre-tassel stage. And so our economic threshold for Western bean cutworm is what we call an accumulated 5% of egg masses. And you're probably thinking, what the heck is accumulated 5%? Well, so say you go into the field twice, you know, pre-tassel, and you count 2 or 3% 1 week, and say you count another 3 or 4% the next week, you have reached that economic threshold of the 5%. So that's why it's called an accumulated economic threshold. The best way to control this, and if you have an issue with this, and if I lived in northern New York growing grain corn, I would definitely try to get my hands on the Vegetative hybrid, because these are the only ones that are really resistant to it. They have, the Western bean cutworm has broke the resistance of most of the rest of the BT traits. So this, if you've had problem with it in the past, if you have a hot spot in your area that has Western bean cutworm, and they kind of prefer that sandy, loamy soil type of situation, you know, it might be a good idea to select this so you're kind of preventing an issue. There are insecticides that you can spray on this if you've reached threshold. And again, it's in that Cornell guide that I mentioned. And they just released, I believe they just released those this week for purchase if you want to get those. And, you know, actually this year might be a good year to do that, just because there's a lot of new information in there relative to seed treatments. We did some new seed treatment tables. I know Jamie Cummings added some fungicide information to the different crops. And then I worked with Elson on doing the insecticide tables. Okay, so that's kind of, I gave you a real quick and dirty biology lesson on those insects. And now I'm going to kind of get into some of the data. So this is our field crop, our field corn pheromone trapping network. There's 27 of us that do it. There we have a mixture of growers. We have a lot of extension educators and crop consultant volunteers that put up a trap just like this, like Mike Davis here in northern New York, puts out traps out on the Worldsboro farm every year for us. And again, so in 2020, we had 20 black cutworm traps across the state. We also had 20 true armyworm traps across the state. And these are probably with, you know, the people that are doing these were pretty much next to each other. So that's probably why we had exactly 20 of each. And again, you start in April and you go through June. And again, we're going to try to extend that Armyworm one later on in the season. And then depending on the year, we've ranged from, 55 up to 70 plus traps for Western bean cutworm across the state. So we have quite the database because we've been doing it since 2010. And again, I like to point out that the corn growers did fund this, Corn and Soybean Growers Association, and we really couldn't have done it without them because these lures and the sort are rather expensive to buy. And it, it is information that we're trying to help you guys get about the flight of these moths and the potential damage that can occur during a season. This is the data that we collected for the last 2 years with black cutworm and true armyworm. And I do find it rather interesting that if you look at the black cutworm data here, I believe these are Mike Stanyard's numbers here and Jody Putnam's numbers, in that they're finding really early on in the spring black cutworm and true armyworm that are coming in on storms. And I went through the data for individual traps, and it would have been kind of impossible to show on a slide, but I corresponded each of those little blurps like this one. And as we go along, and there's a lot of data along here that you don't see in a line graph, but there was a storm associated with almost every one of these. So I do find it, interesting that these are kind of following a very similar trend at least the first 2 years. True armyworm was a little different. And I believe that I had read there was a lot of biocontrol for this insect pest in 2019 in the southern states. And that can be a limiting factor for this insect pest to get up into storms if their populations aren't high before it starts. So that might have been the difference. But they do seem to follow the same trend relative to the storms that do reach to the northeast. This is the specific traps, and these are the season totals. And as you can see, we had some pretty high populations out in Western New York with Mike and Jody's traps. And then we did have one in the Hudson Valley that had a pretty high population of black cutworm. But there were moderate populations across, much of central New York. This next year, we're hoping to get some up here in northern New York. We do had Joe Lawrence did one for us there in Lewis County. But the point is what we do with these is that each week we're monitoring these pests, and our volunteers will put this into a database for us. And I take that data and look at the population for that week. And so if we're getting a population in a week that's over 15 moths in a week at one location, we call that an intense catch. And we can set what we call a biofix date, and we can start calculating degree days. And I'll kind of show you what that looks like in a minute. This is the true armyworm data, very similar. Some of the population was slightly less, but we did have this one trap here that was at 247, which I believe was Jody's trap. And but you definitely were getting some pretty high armyworm populations here in Western New York. So I was talking about that biofix date. So each of these pests, we have degree day models that have been researched throughout the years, and we can calculate when eggs will hatch if we know the date that they're laid. And so we probably don't know the exact day of the week that these moths lay these eggs, but within that week, and I go from the point that the volunteer puts it into the database, we start what we call a degree day model. So for black cutworm, if we take our base temperature of 50 degrees and we start calculating those out, we can predict with relative accuracy when you might start seeing those in the field. And that's the point when we would like you to go look, or your crop consultant go look at those in a cornfield. You have the same for true armyworm. It's not as detailed. I could not find a good degree day model for all the different instars like you do for black cutworm, but I continue to look for that. And so, like I said, this is what we call, I put out alerts. I know that Mike Stanyard puts out alerts, Jody puts out alerts. I know Aaron Gabriel and all the different specialists across the state will put out their own alerts relative to the data they're receiving. So between what I do and what they do, I think we're getting a good information out there on relative when you might start seeing damage relative to the flight of the moths that occur into the state. And so here, there's one here for Seneca Falls. I said it at April 28th. And, and so this is, as we're going throughout the summer, we're at 529 degree days, fifth instar. We're getting towards the end of of those larval stages. So we're able to predict when they're going to occur and when they end. And while it is not perfect, I'm not going to say go in the field, you're going to find fifth instar larvae in Seneca Falls, but you'll probably have, you know, a good chance that if they did survive, if you didn't use seed treatments and the sort, you might start seeing these things. And it might be a good thing to go check to make sure that you're not getting the cut plants and you're not getting that economic damage to your crops. And we did it also for armyworm. So we did it for both. And we put out alerts each week for both. So that's kind of what we did with black cutworm and the true armyworm. With the western bean cutworm, and this is really dramatic this year. It's not as quite as dramatic as in other years, but you look at the numbers. This is the total number of moths caught in traps at each location. So these are 63 locations. These black ones had no catches, which is actually relatively rare. But if you look up here in northern New York, and especially right, you know, along the edge here, along the lake river, you know, some of those populations were at 3,000 moths per trap for the season. I mean, those are intense numbers. And this is kind of a very common thing we see each year. Although we do see hotspots in Western New York and actually some down in Washington County. But, you know, every year, Northern New York is really getting slammed with a lot of Western bean cutworm. So this is the data on what we get on an average per trap number of moths caught. And I have to say, you know, it looks pretty impressive. You go from 2010 to up here to 2017, we go from 13 to, you know, almost 570 moths caught per trap. And I'm going to break this out with Northern New York versus the rest of the state, and you're going to see quite a difference. But, you know, Northern New York is really skewing the rest of the data. But, you know, it is established here. It is spread out across the state, and we're getting significant numbers now. I do like to point out from this point on, 2017, this does not include sweet corn or dry beans. Before that, in 2016, all that data included sweet corn and dry beans, which sweet corn gets fewer moths. I don't know if that's because they spray up more insecticides, but these numbers would reflect higher if you peeled out some of that stuff. And I might try to do that in the near future and see what these numbers actually are. So one of the things that we do with the data is that we want to know when a peak flight will occur. And at peak flight is when most of the egg laying is going to occur and on pre-tassel corn. So if you know in your part of the state where you might be reaching what we call peak flight, it's a good indication that you can go and check shortly after that, you can go scout those fields. And so Dan Olmsted, he's the coordinator of the Network for Environment and Weather Applications with the IPM program, developed this model here that he took the degree day models that were developed through research and then our data that we've collected over 10 years and kind of merged them together into getting us what is a predictive peak model. And what it actually is that, so say here where it says 82%, that means that 82% of the flight has occurred. So much of that peak flight is gone, it's over. Whereas up here, which makes complete sense, you know, Northern New York has 17% of their peak flight has occurred. So they have much of their flight relative to what's going to happen still to come. Now, I do like to point out that we get, it's speculated and seems to be the case, because I could talk about this later. I tried to take soil type and look at the different populations and what soil type that they prefer. But there's a population coming over from Ontario across the lake that's really kind of skewing northern New York's numbers. And so, I mean, they're potential pests too. They're going to lay their eggs on plants, but they are definitely getting a localized population and a migratory population. So, but yeah, this is and we put this out when peak flights are starting to occur for 3 or 4 weeks throughout, through July into early August. And, this is on what we call our field crops pest report, which is through the New York State IPM program. And all this kind of information is there relative to other types of degree day models also for different pests. In the future, we will be working with Dan Olmsted on doing more detailed maps similar to these for other pests like black cutworm, true armyworm, alfalfa weevil, and some of the others. So relative to this last year, which is interesting because it was kind of a hot, dry summer, we had the earliest peak flight we've had in the 10 years we've been monitoring. And it is this blue line right here. And so we were up over on a weekly, this is based on weeks. So this is the week of the last week of July, just before the last week of July. And, that's a per average basis, not a total, per average per trap basis, not a total number of moths caught. So it's interesting as it compares. But if you take this, and I took only the last few years when we started to really get high populations and broke it relative to northern New York versus the rest of the state. Northern New York is just a lot of moths. As you can see here, that you're up over 500, almost up to 460 moths caught per trap. And what's interesting is that the peak flight of northern New York, when I graphed it out this year, was a week later than the rest of the state. And that doesn't occur with all this data, but I think that's going to, as we start to get more data and more traps out, I think that might still, that seems logical because it's warmer. These insects are developing based on heat units. And so that, that's a point of where you're looking at when to be in the field. And that is a good indication of, peak flight is be out there scouting shortly after that. So what's interesting here is we took the data for each year based on the average per trap. And we kind of had some exponential growth up until, till 2018 here. And then it seems that it might be flattening out a bit. And I don't know if, I think we need a few more years of data here to really see if we're kind of upset on what we're going to have each year. But it definitely has a kind of a production function type linear or graph here that, it does seem to be flattening out over time, which would be good. So,
Ken?
also I did take the number that we
Can you hear me?
Yeah.
Hey, buddy, we need to start wrapping up here. We're over time.
Oh, I'm sorry. Okay, let me go to the end here.
Okay.
So basically, what there are significant issues we had in 2020 with true armyworm, especially some with black cutworm. We're going to try to monitor longer into the summer for true armyworm, and then with western bean cutworm, we would like to continue doing these traps. And this is all about helping you guys know when to be in the field, and then also relative to western bean cutworm is that we will be able to monitor that population throughout the state and kind of develop some good degree day models for that. And I apologize, I didn't realize I was over time.
All right. Thanks, Ken. Yeah, no, guys, we're over, but I'm gonna, I'm going last here. I'm the last presenter. Talking about corn nematodes, so I'll go as quickly as I can here. I know we're gonna be over because we got stalled out there with Tony's, but I'm gonna get going here. Can you see that, Brandy? But you can't see me. Okay, how do I show me? Why did I get bumped off of there? Maybe it's not gonna be able to see me. All right, I'll get this shit. Now you can see me. Maybe that's good, maybe that's bad. All right, so corn nematodes. Not a topic I don't think we've ever discussed. I don't hear it being discussed here in New York, but it's something that's been out there for quite a while. So when I was with American Assignment back in the late 90s in Illinois, we dealt a lot with corn nematodes. But I also had a product named Counter, which is a widely used soil insecticide, which also was a nematicide. So there was a lot of work done on nematodes out in Iowa, Illinois, Indiana at that time when things started really coming on board. And a lot of work, a lot of insecticides being put down. So we moved here to New York, really haven't thought much about this. There's always fields that we just can never figure out what is going on in this field. There's a million things that we check and just can never figure it out. And a couple years ago, we had a side-by-side, somebody put out some Nemastrike, which was a seed treatment from Monsanto. And there were some pretty big differences just in the side-by-side. And then Nemastrike got pulled off the market in 2019 and 20. And so I never really went after it again. But we're starting to see a resurgence even in the Midwest. Illinois has been doing a new survey of the state for nematodes the last 2 years. This will be the third year for that. So I said, let's get back into this. Let me take a look at what's going on and relearn some of the stuff I'd learned 22 years ago and catch up with this. Is this something in New York we should be looking at? So what is a nematode? Well, they're microscopic roundworms. And there's a possibility, there's 23,000 species named right now. They think there's 1 million at least of nematodes in the world. And they're all different sizes, different environments from the ocean to livestock and humans. So they're everywhere. About 15% of those are plant parasitic nematodes. So a relatively small volume of those, are feeding on plants. Iowa was kind of the person, the university that went after this first in the 70s and 80s. And into the 90s, they did some surveys, and they found 28 different species of nematodes that were associated with corn in Iowa. Not all those were economic. They probably found a dozen, the dirty dozen, that they found that could be economic in corn. And a few of these, probably 2 or 3 of them, can be very detrimental depending on soil conditions and other stresses. So how do these nematodes feed? Well, the plant parasitic nematodes, when you put them under a microscope, you see this spear-like mouth part there that the arrows are pointing to, and that actually comes out like a hypodermic needle, and it's called a stylet. Think about sort of how a mosquito or an aphid feeds, where they have that hollow tube that once they inject into a plant, or in mosquito's case, your arm, they can inject a fluid. So these nematodes inject that stylet into an individual plant cell, and they can withdraw the plant juices and nutrition from that plant. So looking back at some of the surveys from Iowa here, this dirty dozen I talked about, they got some really cool names. You think of anything that looks like a spear, it's except a spear nematode, but they're dagger, lance, needle, pin, spiral, sting, stunt. They got some cool names, and they're ones you kind of remember. So people doing the naming have done a good job of keeping these kind of current and in our minds. But if you look at these, there's 2 ways that they feed. there's an ectoparasite, which they're feeding from the outside of the plant, like the name sounds, ecto, and there's an endoparasite form, which actually gets into the root system and feeds within the root. Of these 12, these dozen that are known as the ones that can be economic, 9 of those are ectoparasites. So the majority are feeding from the outside of the root, and 3 of them are inside the root. Just a picture here showing, under a microscope, what an ectoparasite looks like feeding into the root there, and what an endoparasite looks like. So, and you see those as it says below. This is a lesion nematode. They have to stain them to actually identify them within the root system because they're so small. So look at some of the symptoms of nematode. How do I know it's even out there? The problem is many will tell you it's a hidden yield loss issue. So it's very hard to look at a field and diagnose, yes, that's nematode injury. One, because you just can't pull the plant up and say, well, there's nematodes. Okay, the perception is reality. We can't even see them unless we have certain laboratory tools. And they can resemble injury from anything. And I'll show you some pictures, but any kind of insects, disease, weed injury, herbicide injury, fertility issues, pH, compaction, it all looks the same. That's what makes it hard to difficult. So I'm going to show you some pictures that are confirmed nematode injury from other states. This one here was from Nebraska, but that could look like a lot of things, right? Could be a lower area that had some water damage, and we had some different pathology issues, some damping off issues in those parts of the field. This one here, we see the plants are extremely stunted and some are missing. Could it be a herbicide carryover issue? Could it be a disease? Could it be some sort of underground insect, wireworm damage, white grub damage? Yeah, absolutely. Somebody showed me this picture. I would tell you that's a common armyworm issue. Plants are missing. Looks like it could be a no-till issue. I've seen this 100 times in the field for common armyworm. Some extreme situations where we have some extreme stunting is pretty obvious, where we see these side by side, and we can see that you dig at the plants and see that is a nematode issue. The other thing is you want to look for is some of these areas that have chlorotic spots in them. And we all see these occasionally. Sometimes they're up on a knoll, but with these rounded circles where the plants just don't do well, they could be stunted as well as chlorotic. And you look up close, it's really chlorotic. Is it a pH issue? Is it a macro or a nitrogen issue? Could be, but these were actually nematode issues. So that was above ground. You go below ground, and again, these are feeding on the roots. So as you dig up these plants, and in this situation, we see that the roots themselves are unhealthy. We see a lot of stunted, clubbed areas, brown areas where this is a primary nematode is a primary issue, but its injury from feeding causes secondary issues with a lot of the the pathology issues that can occur. So there are some alternate hosts that can benefit from this nematode feeding. Up close, again, showing some of this. This could be possible herbicide injury, corn rootworm, white grub. It could be a lot of different things, but this was nematode from lesion nematodes. So there's been some studies done looking at soil preference for these guys. And I mentioned these nematodes are different sizes. There's some that they consider large, which could be, almost maybe a 1/5 of an inch, that put it on that scale, to microscopic. And you look at soil preferences, you see a lot of these have a sandy preference, particularly the needle, the third one down, a very sandy, where they even say it needs, probably above 60% sand. And that's because that's the largest nematode out there. And it needs those large pore spaces to move throughout the soil to get to the roots and go plant to plant. As we move to some of the smaller ones, it's non-specific. The pin, the spiral, clay loamy, stunt just says soil. So as they get smaller, they don't need that large pore size. And just to think, I said needle's the largest one. And I remember some of the visuals I had when I was a tech rep is I used to have some needle nematodes in a vial of alcohol. And I would actually, you couldn't see them, just looked like vial alcohol. But when you shook it up like a snow globe and you held it up to the sun or to the light, you could see these microfibers. I always thought of them as like asbestos fibers floating around. And there was hundreds of them in this vial. And that's how small the biggest one is. So just to kind of give you some perspective. So what about thresholds? The title can these reduce yields? Well, yes, they have been known to reduce yields, but it really depends on which species. If you look at this threshold, and this is per 100 centimeters cubed which is just under a half a cup of soil. To put that into perspective. Some of these, like the needle, you can see it's only one, per 100 centimeters cubed, as well as sting. So those ones, you find one in that sample, it's high, and that is threshold. Some of these, you get the dagger lance at 30 to 40 to 60 or in that moderate range. And you have some of these ones like spiral, you can have 500 to 1,000 in that sample and then, still be under that threshold. So again, it really depends which one you have and relatively their size. So is it an economic pest? And this was really hard to find in the research looking around to find any kind of studies from economics. There was one statement, and you couldn't even trace it back to a research project that said in 1994, it was estimated that corn yield losses due to nematodes were valued at $81 million in Illinois alone. And I couldn't find out who said that. But the nematologist that came to Illinois after that, Terry Nieblek, says that, since that time, she knows that it's increased dramatically because of three things that have happened since 1994. The first one there being continuous corn, at least in the Midwest, more continuous corn. Not that it's an issue here, depending on what your definition of continuous corn is. Is it more than 3 years, which, most of our silage can be 3 up to 4 years. The second was use of GMO, so BT corn. And I was out there in 98 when the first BT corn was being planted for corn borer and for corn rootworm. And I mentioned before, counter was a big product that I was, using at the time. And so the reduction of seed insecticides at planting because of BTs, they said is leading to more, less management of these nematodes while we were using insecticides like counter. And the last one is increase in no-till or conservation tillage acres, which, these pests like earthworms, you think of them as very, very, very small earthworms, they're not related, but, they like undisturbed soils. You think about what was there, before we started farming, it was these grass prairies, and they dramatically did well in these grass prairies. And the problem with a lot of these nematodes is that they can feed on multiple species. Okay, so ones that feed on corn can feed on grasses, small grains, and even soybeans. So in many cases, they're not real specific. I looked up, what about statewide corn nematode surveys that have been done? What are they seeing? And so most of the Midwest states have done work like this here, at least in the 2000s. I mentioned Iowa's that I showed a lot of data from already. Illinois did a lot of stuff in 08 and 09, and found that of those 2/3 of those fields with lesion nematodes were at risk for yield reduction. So high yield reduction, looking at spiral lesions, stunt and lance nematodes. Indiana at Purdue, they mentioned needle nematode was there, probably under sandy soils was #1, but they also had root knot, lesion, dagger, lance. Ohio did a survey in 13 and 14, over 400 fields, found plenty of nematodes. And what they found interesting in some of these fields, they found 9 different species, not just one species. It was from 4 to 9 different species in the same field. And I think we'd see that in most of these surveys. Pennsylvania, they weren't looking in corn, but recently in 2018, they were looking in soybean for a soybean cyst, and pulled out lesions, stings, spiral, stubby root, lance, and dagger in soybean fields. Like I said, and not just corn, these ones also feed on soybeans, which makes them more difficult to go after with rotations. So how do we sample for these guys? Well, it's not much different than a soil sample, except that we want to kind of angle down in underneath that and get those root pieces in our soil sample. Ideally, most of these saying between V4 and V7 is the best time to be sampling for corn nematodes. So before reproductive, before our soils get real dry in the upper layers, 8 to 12 inches, 20 cores per field. But also if you see fields that you actually see injuries you can't identify as anything else, you think it could be nematode, they want 4 to 6 of these plants sent in with that. So again, it's not hard to sample these and get some good results. And there's some good labs around the area. So what are some current management situations that we can do to manage these nematode populations once we identify that we do have nematode populations? Nothing can be done in season. Okay, we don't have anything systemic that we can apply to the plant to get down to the roots and take care of it. In some cases, rotation, but you really have to know what species and look up, what are its preferred hosts and manage that. It may be an alfalfa, it may be a clover, a legume of some sort that it's not known to feed on. Soil applied counter as an insecticide plus a nematicide. if that's still found. I haven't seen counter in quite a while. But mainly right now we're looking at seed treatments. Now, up to about 2010, there was only about 2 of them that were being used. And most of these, if you look down at Avio and Lumialza, Botivo, and Chunemco, that's how you say it, they're microbial. So a lot of them are bacteria that are used to colonize the roots and kind of form a barrier to keep out these nematodes. Some say that they're a paralyzing factor. Others, they're just saying, well, we're providing more nutrients and more water and less stress to these plants to combat that nematode damage. So that seems to be the route people are going with these biologicals with their seed treatments. And some of these are very specific by company, whether it be Pioneer or even Beck's. But there are a couple that are still insecticide nematicides. Avicta is abamectin, which we've been seeing more with some of the spider mite control as a good product. The Nemasect, again, is an insecticide nematicide mix. As a Nemastrike. Now, Nemastrike, at least of last year, was off the market, but it was the only product that I've actually seen here in New York that showed some promise just on a side-by-side, not replicated. So I think there's some more products out there that you're going to see that are going to be applied already to the seed and have some potential. So what am I going to do with this whole thing with corn nematodes? I think it's something that needs to be looked at here in New York. As far as I could tell, I couldn't see any surveys. There may be some industry folks that maybe have taken some samples I don't know about, love to hear about it, what species were, but the main thing for us right now is what species do we have present in New York? We have all sorts of soils, again, from the mucks all the way to the gravels. Is there a certain species that is favorable here in New York? I'd love to know. And if we figure out the species, are we seeing damaging population numbers, or are they low enough that they're really not an issue unless maybe they're combined with other stress factors? I really want to start looking, for ID fields with injury symptoms, and I'd like to do some sort of sampling program here in Northwest New York, in our 9-county region here, this summer if I can. Going to look for some grant monies. In samples about $40 to $50 to get them done and surveyed and do some species work. So I'd love to start with 50 to 60 samples here in these 9 counties. And just to kind of get an idea, what do we have? What numbers do we have? And is there something we need to do or not? And I think those questions need to be answered. And I'd like to kind of spearhead that this year. So that's really quick. I was just trying to catch us up a little bit. I know we're over, but that was just really quick on what we're doing. And that was it for me, folks.